Good day everyone. I am Mr. Ishitu Tasfaye, your biology teacher. This is grade 12 biology, week 14. The lesson for this week is chapter 35, lesson 3, fighting infectious disease. Your textbook page 1020 to 1023. Objectives of this lesson. At the end of this lesson, you should be able to distinguish between active immunity and passive immunity, describe how public health measures and medications fight disease, and finally, describe why patterns of infectious disease have changed over time. In today's lesson, which is fighting infectious disease, we are going to see three major sections. These are acquired immunity, public health and medication, and finally, new and re-emerging disease. Let's get started with the first one, which is acquired immunity. What is acquired immunity? Acquired immunity is a type of immunity that develops during a person's lifetime. This immunity doesn't exist at birth, rather acquired from the environment through infections or vaccination, or by transfer of antibodies from donors to recipients. How do vaccines and externally produced antibodies fight disease? A vaccine stimulates the immune system with an antigen. The immune system then produces memory B cells and memory T cells that quicken and strengthen the body's response to repeated infection. On the other hand, antibodies produced against a pathogen by other individuals or animals can be used to produce temporary immunity. Let's look at the historical development of acquired immunity. Some 200 years back, an English physician by the name Dr. Edward Jenner performed an experiment in which he put fluid from a cowpox patient's sore into a small cut he made on the arm of a young boy named James Phipps. As expected, James developed mild cowpox. 2 months later, Jenner injected James with fluid from smallpox infection. However, the boy didn't develop any kind of smallpox. The boy's cowpox infection had protected him from smallpox infection. This injection of a weakened form of a pathogen or of a similar but less dangerous pathogen to produce immunity is what we call it vaccination. The term comes from the Latin word vacca, meaning cow, as a reminder of Jenner's work. There are two types of acquired immunity. These are active immunity and passive immunity. Let's see what an active immunity is. Active immunity may develop as a result of natural exposure to an antigen, fighting an infection, or from deliberate exposure to the antigen through a vaccine, for example. Vaccination stimulates the immune system with an antigen. The immune system produces memory B cells and memory T cells that quicken and strengthen the body's response to repeated infection. Now, the second type of active immunity, which we call it passive immunity. Antibodies produced against a pathogen by other individuals or animals can be used to produce temporary immunity. If externally produced antibodies are introduced into a person's blood, the result is passive immunity. Passive immunity lasts only for a short while because the immune system eventually destroys the foreign antibodies. Passive immunity can occur naturally or by deliberate exposure. Natural passive immunity occurs when antibodies are passed from a pregnant woman to her fetus across the placenta or to an infant through breast milk. For some diseases, antibodies from humans or animals can be injected into an individual. For example, people who have been bitten by rabid animals are injected with antibodies for the rabies virus. Now let's look at the second major section of this lesson, which is public health and medication. How do public health measures and medications fight disease? Public health measures help prevent disease by monitoring and regulating food and water supplies, promoting vaccination, and promoting ways that avoid infection. On the other hand, antibiotics can kill bacteria, and some antiviral medications can slow down viral activity. In 1900, more than 30% of diseases in the United States were caused by infectious disease. However, some 100 years later, only less than 5% of diseases were caused by infectious disease in the United States. Two factors that contributed to this change were advancement of public health and the development of medications. Public health measures. In 1854, Dr. John Snow learned through interviewing residents and mapping that the source of a London cholera outbreak was a water pump. This is a major event in the history of public health. The field of public health offers services and advice that help provide healthy conditions. Promoting childhood vaccinations and providing clean drinking water are two important public health measures that have greatly reduced the spread of infectious disease. Medications. Medications such as antibiotics and antiviral drugs are other weapons that can fight pathogens. An antibiotic is a compound that kills bacteria without harming its host. In 1928, Alexander Fleming noticed that a mold by the name Penicillium notatum seemed to produce something that inhibited bacterial growth. Research determined that this something was a compound. Sir Alexander Fleming named it penicillin. Researchers learned to mass produce penicillin just in time for it to save thousands of World War II soldiers. And antibiotics such as penicillin have no effect on viruses. However, antiviral drugs have been developed to fight certain viral infections. These antiviral drugs generally inhibit the ability of viruses to invade cells or to multiply once inside cells. Now to the last section of this lesson, new and re-emerging diseases. Why have patterns of infectious disease changed over time? Two major reasons for the emergence of new diseases are the ongoing merging of human and animal habitats and the increase in the exotic animal trade. Misuse of medications has led to the re-emergence of diseases that many people thought were under control. By 1980, many people thought that medicine had conquered infectious disease. Vaccination and other public health measures had wiped out polio in the United States and had eliminated smallpox globally. Antibiotics seemed to have bacterial disease under control. Some exotic diseases remained in the tropics, but researchers were confident that epidemics would soon be history. In recent decades, however, a host of new diseases have appeared, including AIDS, SARS, hantavirus, monkeypox, West Nile virus, Ebola, avian influenza or bird flu, to mention some. And what about COVID-19 right now as we speak, which infected more than 1.6 million people worldwide and resulted in more than 100,000 deaths globally. Other diseases that people thought were under control are re-emerging as a threat and spreading to new areas globally. Changing interactions with animals. As we have said earlier, changing interaction with animals is one of the causes for new and re-emerging diseases. Pathogens are also evolving in ways that enable them to infect different hosts. As people clear new areas of land and as environment change, people come in contact with different pathogens. Exotic animal trade for pets and food has also given pathogens new opportunities to jump from animals to humans, which we call it the zoonotic disease. In 2003, for example, dormice from Ghana spread monkeypox to prairie dogs in the United States, which in turn infected humans. The spread of SARS also has been associated with the wild animal trait. Misuse of medications. Misuse of medications has led to the re-emergence of diseases that many people thought were under control. For example, many strains of the pathogens that cause tuberculosis and malaria are evolving resistance to a wide variety of antibiotics and other medications. In addition, diseases such as measles are making a comeback because some people fail to follow vaccination recommendations. Now let's answer the following lesson check questions. Question number 1. Explain how vaccinations and externally produced antibodies help the immune system to fight disease. The answer: vaccination stimulates the immune system with an antigen. The immune system then produces memory B cells and memory T cells that quicken and strengthen the body's response to repeated infections. On the other hand, externally produced antibodies, which are antibodies produced against a pathogen by other individuals or animals, can be used to produce temporary immunity. Question number 2. Describe the difference between active and passive immunity. Well, active immunity may develop as a result of natural exposure to an antigen or from deliberate exposure to the antigen, while passive immunity occurs when externally produced antibodies are introduced into person's blood. Question number 3. What are the goals of public health measures? Sample answers. The goals of public health measures is to prevent disease by monitoring food and water supplies, to promote vaccination, and to recommend ways to avoid infections. Question number 4. Why is it important to discern if a sickness is caused by a bacterium or a virus? This is because antibiotics can kill bacteria, but they have no effect on viruses. Question number 5. List 2 major contributing factors involved in the spread of new and re-emerging diseases. The 2 major contributing factors involved in the spread of new and re-emerging diseases are changing interaction with animals and the misuse of medicines. And finally, number 6, how do you think the ease of global travel has affected the spread of emerging diseases? Explain. And the answer is, global travel has increased the spread of emerging diseases because infected people who show no symptoms can travel around the world very quickly, spreading the disease more widely than possible in earlier times. Thank you for watching and see you very soon.